Hi, everyone. My name is Beth Roskilly, and I am a PhD candidate in Sally Aiken's lab at the University of British Columbia. And today I'm presenting work from my thesis, and I'm studying range-wide patterns of climate adaptation and phenotypic plasticity in Western larch. And this work is part of the larger co-adapt project. So Western larch is one of the few deciduous conifer species native to Western North America, and it has a relatively narrow distribution depicted here on the map that spans about 7 degrees in latitude and 1,500 meters in elevation. Climatically, that is a range of about 5 degrees Celsius mean annual temperature and 1,500 millimeters mean annual precipitation. Previous common garden studies have shown that the phenotypic clines are of relatively modest slope compared to co-occurring conifer species or conifer species that span a greater climatic gradient, and that there's lots of variation within populations, suggesting high phenotypic plasticity and high standing genetic variation. In BC, Western larch is the 5th most planted species, and species distribution modeling projects that there's going to be a large expansion of areas of suitable climate in BC for Western larch. And that's depicted here on the map on the left, showing seed planting zone expansions for 2030. The future zones in an expansion in the yellow and the blue relative to the current breeding seed planting zones in the green. And larch has also been the first species to be planted operationally outside of its range and restricted to about 10% of trees in a stand. So it's this candidate for testing the ideas around assisted migration, and this serves as incentive for the objectives of my thesis, which are to characterize patterns of local adaptation using phenotype and genotype environment associations, and identify the climate drivers and the putative loci underlying adaptive plastic and genetic responses. I'm also interested in using genomic prediction to estimate future adaptive potential of populations, and another main objective is informing future management and breeding strategies for larch. We are including breeding material from the BC breeding programs in this research. The sampling for this project is range-wide, and there's basically 2 groups of data. There's the common garden experiments that I'm doing out at UBC. That's represented, the location is here, represented by the yellow star. And genomic data collected from provenance trial in Southeast BC, shown with this yellow triangle. There are 52 natural populations in the common gardens in the blue and the purple, and 40 natural populations that were sampled from the provenance trial in the red and purple. So the purple represent the 32 populations that overlap between the common gardens and the provenance trial, for which we'll have phenotypic and genotypic data. And then also included in the common gardens are 28 breeding families from the 2 breeding zones in BC. So last year, we managed to plant out 1-year-old seedlings that were going in the greenhouse at the beginning of the season in spring, just when COVID hit. And we measured height growth throughout the season once they were planted in these outdoor beds. And I also measured soil moisture and midday water potentials, as I'm interested in drought tolerance, and I've been implementing drought treatments in some of the beds. We recorded bud set phenology, and I measured fall cold hardiness and recorded leaf senescence phenology in the fall. So today I'm going to be presenting the fall cold hardiness data because it is the furthest along in analysis. And we sampled 40 natural populations from the control treatments only, so well watered. And we used branch segments of about 15 millimeters in length at each sampling. And we measured electrolytic leakage, measured at 3 different temperatures, control 4 degrees C, a -15, and a -21. And this was over 2 weeks in late October. The data I analyzed and I'm presenting in this talk are from the first week at -15 degrees C and the second week at -21 C. So here's the data, the cold injury values plotted by provenance, and going from the least cold injury to the most cold injury. And the raw data is what's in light blue, and then on top of that in the black are the estimated means per provenance. And what you can see with this plot is that there is high variance within populations, and there's only about 14.6% of the variance is explained among populations. In terms of climate predictors, one of the strongest climate predictors for fall cold hardiness in the other species we've studied in the lab is provenance mean coldest month temperature. And so that's plotted here with average cold injury. But as you can see, the relationship is not strong, and it doesn't explain a lot of the variance. These are preliminary data analyses, and there's more to be done, but so far we're not detecting much of a strong relationship. Looking at the geographic trends in cold injury, we do see a trend of less cold injury in the northern populations and high elevation southern populations. So what's shown here is the northern populations are in the dark blue and the southern populations are in the lighter blue. And the northern populations tend to have lower cold injury compared to the southern populations, except for the high elevation southern populations that have similar cold injury values. There's a lot of spread to this data though, and this model only explains about 15% of the variation. The R-squared is fairly low. So here's what that data looks like when mapped with lower cold injury values in the darker colors, also meaning darker can also be thought of as more cold hardy. And so you can see there's a greater concentration of darker blue points in the northern portion of the range compared to the southern portion of the range, where you see more variability and following a little more dependent on elevation. So the conclusion so far is that we're not seeing that Western larch is as strongly locally adapted to cold temperatures as we've seen in other species that span a larger climatic gradient. I do want to follow up with more cold hardiness data this fall and include some of the breeding families for comparison. We're also interested in if late spring frost is a stronger driver of local adaptation in Western larch, given that it's deciduous. And I have freshly collected bud break data from this spring that I wish I had analyzed for this talk, but that we will be able to use that to test that hypothesis. And then we'll also be looking at local adaptation to drought using the drought experiments that we will implement this summer. And then we still have lots to do with the genomic data, which is pooled exome sequence capture data, and we'll be doing genotype by environment associations. So you'll have to stay tuned. And I just want to make sure to acknowledge all those who helped make these common garden experiments possible and manageable, and also the funding sources and the seed, those that provided seed as well. And so thank you very much. I am looking forward to questions and the rest of the symposium.
Hi everyone. Thank you so much for having me here today. My name is Kate Peterson, and I'm here to talk to you a bit about research I've been working on as part of my PhD at the University of British Columbia. Myself and my co-authors have been working on this project for a few years now, and the title is Predicting Lodgepole Pine Mortality in a Changing Climate Using Provenance Data. This project arose out of a need to examine the ability to manage British Columbian forests for climate change impacts. The impact of projected climate change is still unclear for many tree species, and additionally is likely to vary throughout the range of all species and in different populations. And a major issue in forestry in BC and in Canada is that most models used to predict forest growth and yield are not climate sensitive. And this means that we lack the ability to accurately simulate forest stands over time under different climate conditions. So we chose to focus on one species for the first step of this project, and we chose lodgepole pine as it's an ecologically and economically significant species in the province of British Columbia. Our objectives of the study were to link lodgepole pine mortality with climate, and that's climate of the planting site or test site and climate of the provenance, which is where they originate. We wanted to predict stand level percent survival, as well as the probability of survival of individual trees in a stand as a function of climate, and incorporate these models into a growth and yield model so that we can simulate climate change impacts over time and have a better idea of what climate change may look like on BC forests. So speaking of growth and yield models are used to simulate forest growth over time and project things like timber yield on the landscape. Growth and yield models are usually either stand level or individual tree level. So stand level estimates provide things like volume per hectare, and individual tree models will typically give us stand level estimates as well as individual tree estimates like the biomass or height or diameter of an individual tree. So one of the objectives of this project was to develop our methods in a way that you could incorporate in many different types of models and had the flexibility to be applicable in both. So the data for the study comes from the Illingworth trial, which is a wide-ranging long-term provenance trial that took place mostly in the province of BC, but also up into the Yukon. You can see the test sites there are the red squares, and they cover most of the province of British Columbia, and the seed sources came from most of the natural range of lodgepole pines. You can see they stretch down from Southern California all the way up almost into Alaska. The climate data that we used for the study comes from Climate BC and Climate NA, and the sites were repeatedly measured. So they were measured 6 separate times. So the latest is year 32. And the study was initiated in 1974. So our first step was to fit a transfer function. And this is just a general explanation of transfer functions. So transfer functions are functions that relate growth variables, such as height or mortality, to climate transfer. And climate transfer is defined as site climate minus provenance climate. So this means that a positive transfer distance means that a seed source is moved to a warmer climate, and a negative transfer distance means a seed source was moved to a colder climate. And transfer functions are very useful in allowing us to examine the possible impacts of climate change. We can use transfer functions on a single site, for example, and apply future climate predictions and predict how climate change may affect the populations already growing on that site. And transfer functions can also be used to determine what seed source may grow optimally at a certain site. Results of our transfer function. Our transfer function predicts stand level stems per hectare. So it's meant to adjust the predicted stems per hectare in a growth and yield model based on climate projections or differences in climate. So it predicts stand level stems per hectare as a function of mean coldest month temperature, mean summer precipitation, as well as top height, which is the average height of the 2 tallest trees in a population. We included top height as a continuous variable in this model because it allows the function to be dynamic as the trees grow, which is a really important step. Most transfer functions are usually fit on a snapshot of measurement. So it's a single year, and it has to rely on the assumption, if you want to do simulations or look at any other time in the tree's life cycle, it has to rely on the assumption that the relationship between climate and mortality is constant throughout the life cycle of the tree, whereas it's very possible that this isn't the case. So we included top height as a continuous variable so that we can look and make predictions for any height of the trees in a stand. We also included both site and provenance climate, so mean coldest month temperature, mean summer precipitation, as inputs, because this allows our function to be population specific. So we can get different predictions for stems per hectare depending on the climate of the population or the seed source. So the results of our transfer function, it shows that the lodgepole pine response to mortality is response to climate is in fact population specific. You can see that the coldest population, or the population from the coldest provenance, the solid black line, it would see slight increases in survival under climate warming. So you can see that the vertex of the curve is slightly to the right of 0, suggesting that a slight positive transfer, so moving to a slightly warmer climate, would cause small, or would be associated with small increases in survival. This isn't true for any of the other populations examined in these graphs. You can see that populations from warmer provenances would likely see decreases in survival from any positive transfer, so any climate warming. This is pretty significant when it comes to climate change science, because this basically shows that all populations will likely see decreases in survival under climate warming. Even the coldest populations eventually, it's just depending on the degree changes depending on how cold the population climate is. So the next step of our research was to fit an individual tree probability function. And that our goal was to predict the probability of survival of each individual tree in a stand. And this was done so that we could take our stand level results and downscale them into an individual tree model, and therefore select which trees to kill to get to our predicted stems per hectare. So our individual tree probability function uses height increment, relative height, and mean summer precipitation as inputs, so the growth per year and the height relative to the other trees in the stand. So you can see there on the left-hand side, the graph is showing two different height increments and how the probability of survival over the relative height of the tree. So you can see that trees with smaller height increments, so that are growing less per year, are more likely to die due to climate, to be associated with climate-induced mortality. The applications of this research are intended to be flexible. So the annual estimates, as well as the two-step process with the two separate models, allow for greater use and greater applications. We can look at stand level growth and yield models, look at mortality in those models just using the first step, the transfer function, or we can utilize these results in individual tree growth and yield models where we actually need to select which trees to kill. So the goal would be to use the stand level transfer function to estimate the number of stems per hectare in a stand per year, and then use the individual tree survival probability function to select which trees to remove each year in the simulation. So this graph included in this slide is just an example of what a simulation would look like using the stand level transfer function. You can see that it's mean coldest month transfer, and the solid black line is a population that hasn't been transferred, and the dotted line is a population that's been transferred to a site that's 2 degrees warmer. And you can see that that leads to a decrease in predicted stems per hectare over time. So in conclusion, lodgepole pine response to climate is population specific. But that being said, lodgepole pine is likely to see widespread decreases in survival over time under continued climate warming. And as you can see by the graph on the right, climate warming is projected to increase over the next 100 years in British Columbia. Additionally, climate-adjusted simulations can help us understand possible climate change impacts on lodgepole pine. So this research is really important because we can use the results of these functions in growth and yield models and therefore simulate lodgepole pine stands over time and determine what some of the impacts of climate change may be. And thirdly, the methods developed here can be used for other species and other jurisdictions. So our goal is to move forward with this research by fitting similar models on other species and looking at height as well. Anybody have any questions?
Hello, I'm Juliette Archambeau, and I'm currently doing my PhD under the supervision of Santiago González Martinez and Marta Benito Garzón at the University of Bordeaux in France. And today I'm going to present our work on the use of genomic data in predictive models of trait variation at large geographical scales. For conservation and breeding strategies, we have to assess the potential of populations to accommodate future climatic conditions. In forestries, a long history of common gardens has provided a unique framework to associate quantitative trait variation with large environmental and geographical gradients. For instance, here, the population response functions for height along a gradient of mean annual temperature. This framework has allowed to separate the plastic and genetic components of trait variation, identify their potential underlying drivers, and predict trait variation under climate change scenarios. However, this framework relies on phenotypic data from common gardens, which are expensive and time-consuming to maintain. Integrating genomic data in predictive modeling could allow to better understand the adaptive and neutral genetic processes resulting in adaptive trait variation, provide a mean to include the intraspecific variability at a finer scale than current predictive models, adopt predictive frameworks for species and populations for which phenotypic data from common gardens are not available, and increase the chances of fast adaptive decision-making in forest conservation strategies. Different ways of integrating genomic data in predictive modeling of adaptive trait variation have been explored. For instance, Mahoney et al. Identified sets of SNP positively associated with a phenotype, which they called Ps for positive effect alleles. And then they calculated the number of Ps that each individual has. They showed that the patterns of phenotypic variation were equally or better explained by the Ps than by the climatic variables. For instance, here the positive effect alleles, represented by the dark solid line, explain the phenotypic variance on the y-axis, as well as the climatic and geographical variables. Genomic tools therefore hold great promise for predicting future variation in adaptive traits. However, a robust framework to make reliable predictions across species has yet to be established. We can therefore wonder, first, how best to integrate genomic information into range-wide adaptive trait prediction, and what is the added value with respect to current modeling approaches, especially the population response functions I presented just before. Second, an essential component of improving the predictions of population future responses to climate change is to understand the contemporary patterns of adaptive trait variation. And so we can wonder, what are the underlying drivers of the genetic and plastic components of adaptive trait variation? And finally, if we want to predict the population responses at large geographical scales and for a large number of species, we can wonder whether genomic data could be used to predict trait variation at large geographical scales when common garden data are not available. That's the main questions we tried to answer in a case study in maritime pine, in which we used genomic data to improve the prediction of height growth variation across the species range. Here is the clonal common garden experiment we used. Trees are planted in 5 sites under contrasted environments and come from 34 provenances from the 6 known gene pools in maritime pine, represented by the pie charts on the map. We had more than 33,000 height observations, climatic data for the test sites and the provenances, and the trees were genotyped for about 5,000 SNPs. So with the genomic data, we calculated counts of height-associated positive effect alleles for each tree. So first, using the genomic data and phenotypic data from the 5 common gardens, we ran a GWAS to identify the 350 SNPs that show the highest association with height growth at large geographical scales. And we counted the number of positive effect alleles for each genotype that we call the global Ps. Second, we did the same but taken separately the phenotypic data from the Iberian, the French Atlantic, and the Mediterranean common gardens. We ran 3 regional GWAS to identify the SNPs that show the highest association with height growth in a particular geographical region. And then we counted the number of Ps for each tree given its planting site that we call the regional Ps. So we had climatic, genomic, and height data. And from the genomic data, we knew the proportion of belonging to each gene pool for each genotype. And from both the phenotypic and genomic data, we had counts of regional and global positive effect alleles. With that, we first aimed to quantify the relative contribution of the genetic and plastic components of height growth variation and their interaction. And for that, we used the standard quantitative genetic models, which only rely on phenotypic data from the common gardens. So this standard model shows that the mean height value of the provenances on the left, standing for the genetic component, are lower than that of the test sites on the right, which stand for the plastic component. And in fact, in this model, the plasticity explains more than twice height growth variation than the genetic component. This was expected, as in forestry common gardens, the plastic component of quantitative trait variation is generally higher than the genetic component. So secondly, we aim to detect association of height growth variation with potential underlying drivers of the plastic and genetic components. Briefly, we showed that the plastic component of height growth in maritime pine is likely to be driven by multiple and interacting confounded drivers, including the climate, but probably also the biotic environment, soil quality, and other factors not measured in our study. Population structure was an important driver of the genetic component and was associated with adaptation to climate for most provenances. However, there is clear evidence of the importance of adaptation to climate in maritime pine. For instance, some provenances come from the same gene pool but have very different growth rates associated to differences in altitude and or climate. So last, we aim to predict height growth of unknown provenances with models that do not rely on the common garden design, but that include the population structure, climatic variables, and the global and regional Ps. Here is the comparison of the model predictive ability specific to each common garden and evaluated by the variance explained on 6 provenances that were not included when fitting the models. Models combining climatic and genomic data in green predict height growth better than models relying only on the common garden design in red orange, suggesting that models integrating climatic and genomic data better capture the data generative process behind the genetic component. Differences between models were particularly high in Madrid and Cáceres, the two sites that experienced severe drought when the trees were young, and models including the regional positive effect alleles have a better predictive ability than models including the global positive effect alleles. So to conclude, I would say that the most important contribution of this work are the improved height growth predictions for new provenances when combining the population structure, the provenance climate of origin, and the height-associated positive effect alleles. The particularly higher predictive ability of models in integrating climate and genomic data, in particular in harsh environments, and the higher predictive ability of positive effect alleles that show an environment-specific association with height growth, which suggests a predominance of conditionally neutral alleles, so alleles which are adaptive only in particular environments. Thank you for your attention.